All right, we'll go ahead and get started. Few announcements. Last class that audio feedback was driving me and came in on Friday to try to work it out, and it wasn't doing it. So still doing it a little bit. I don't know if it's the microphone. I'm not sure what it is. It doesn't seem to be quite as bad. No, it's still doing it. Anyway, I'll keep working on that. I haven't figured out what's causing that. So a few announcements. There are a number of you, like 10 to 12 of you, that I noticed you have gone into the mastering website and you haven't done anything. A student alerted me to the fact that none of the assignments were showing up on her mastering. It's because you have to turn off the pop-up blockers. So if you're logging into mastering and it's just a blank sheet, you've got a tab on the side and you're not seeing anything else, it's because you need to turn off those pop-up blockers for that website. You might have done it to begin with when you first got into mastering, but for that particular address. Okay, so make sure you do that. If you're not seeing any assignments, there's something wrong on your end, you have to just Google how to do that, and it's not too hard. So write to me if you need an extension for that first question that you do on Friday. All right. Remember, I will not be here on Wednesday, so I look for a recorded lecture. I'm hoping to do that tomorrow afternoon, maybe tomorrow night at the latest. If I'm hoping to do that, so look for that. I'll send out an email tomorrow just reminding you not to come. I hate to do that, but it was just, again, unavoidable. So I'm planning on whatever I don't get through today, including through chapter 6 on the exam that's a week from Wednesday. So hopefully I'll get into the Eukaryotes a little bit today, and then through chapter 6, that's nutrition and growth. And then I'm planning on next Monday starting metabolism, but that won't be on your first exam. Okay, so I'll post something regarding that, exactly how much material is going to be on that first exam. Okay. Think about, I'm planning on everybody taking the exam here next Wednesday. If you have a special circumstance, there are a few people that are basically taking the class online. Write to me just so I can start making a list. The people that I said you could come and take it in a smaller classroom setting. Just write to me if you have some special circumstance. I'm really trying to keep it to only a few people actually taking that exam by respondus. Just to keep the integrity of the test, I want people to be doing that in person. So start thinking about that again if you have a special circumstance. And lastly, there are a couple of questions on that chapter 1 homework that relate to spontaneous generation. I thought that I had gotten rid of questions that I wasn't going to be covering in class, but I missed a couple. So sorry about that. Just look those up in the book. Anything about spontaneous generation won't be on that first exam, and that was back kind of in chapter 1 history stuff. Okay. All right. So where we left off last time, we had talked about some morphologies of cells, cell arrangements. We've gotten into special structures of prokaryotic cells. So we were talking about flagella structure and fimbriae and cell membranes. We didn't get cell membranes yet. We're going on the outside, moving in capsules, like calyces. And we kind of finished up here with this graphic comparing the 3 different kinds of cell walls. And I left you with the question, if you have an organism of each of these kinds of cell walls, what's the hardest to kill with antibiotics? Okay. I don't know if you gave this any thought or not, but the mycobacteria are definitely, from most difficult to easiest. The mycobacteria are going to be the hardest to kill. We talked about this component of the mycobacteria cell wall. Remember, we call these acid fast. It's an acid fast cell wall. That mycolic acid gives that cell wall kind of a waxy appearance. So basically, a lot of our antibiotics don't penetrate through that mycolic acid. All right, so we only have a very small repertoire of organisms that work against, or antibiotics that work against mycobacterium. Probably the next hardest to treat is not a lot of people see this thick peptidic glycan and they say, oh, antibiotics can penetrate that. But remember, it's kind of just a meshwork. It's not semi-permeable like a cell wall is. So actually, it's the gram-negative that are harder to treat than gram-positive because they have this extra outer membrane. So basically, two membranes for the antibiotics to penetrate through rather than just one, the cell membrane. Okay, so that just makes those gram-negatives, again, a little bit more impermeable to antibiotics. A gram-negative infection is always a little bit harder to treat. Again, we just have less antibiotics to choose from there. Not that gram-positives are easiest, are easy in any sense of the way, but maybe easier than these other two kinds of cell walls. All right, so we spent a lot of time on the cell wall. We'll keep working our way in to the prokaryotic cells. We're gonna just talk through the cell membrane kind of briefly here because bacterial cell membranes are very, very similar to our cell membranes. So I assume you've seen the fluid mosaic model at some point before. Function of that cell membrane is just to be, again, a semi-permeable barrier. It allows some things through, some things not. It maintains a concentration gradient. This will be really important when we get to talking about metabolism and aerobic respiration. You have to, maintain the proton ion concentration is high outside the cell, and then they only allow those ions to come in one part of the cell membrane, and then that triggers a phosphorylation of ADP to ATP. So really important to keep certain things out. Again, important in energy reactions. Okay, so we got some proteins mixed in there. A lot of those proteins are important for transport. So if we think about transporting things in and out of the cell, if that doesn't require any energy expenditure by the cell, then we call that passive transport. Okay, so passive processes. All right, of course you know about diffusion. That's a movement of materials from a high concentration to a lower concentration. What kind of things are going to enter the cell membrane by diffusion? Really small things like water, like simple sugars, certain ions, anything that's going to be soluble in lipids, like maybe an alcohol can get through by simple diffusion. Sometimes the diffusion of something requires a special protein channel, so we've got some, we call that facilitated diffusion if it requires a protein channel, and that can be, specific or non-specific. Osmosis is diffusion of water. All right, I want to review osmosis just because this is one of those big principles of biology that you just got to know. I find students that get confused about this all the time. There will be some question on the exam about which way the water flows and what kind of circumstance. All right, so these terms, isotonic, hypotonic, and hypertonic and hypotonic, they're relative terms. So you would never say, oh, that solution is always hypertonic. Well, hypertonic compared to what? Okay, so these terms are I think what people get confused on. These terms always are going to describe the medium that the cell is in, not the cell itself. Okay, so think about the chemistry of the medium could change fairly rapidly. For the cell chemistry isn't really going to change all that much. So these terms always describe the solution that the cell is in. And it's describing the amount of solutes, so something that doesn't dissolve in that medium. So salt, for example, or maybe sugar, for example, something that can't readily pass through that semi-permeable membrane. All right, so if there's a higher amount of solutes outside of the cell, we say that's a hypertonic solution. And you could just memorize that that makes water leave the cell, but it's even better to think through it. Okay, if you've got high, let's say a concentration of salt. So when I put brackets, that means concentration outside of the cell. Okay, if the solute concentration is higher, then you're going to have a low concentration of water outside of the cell. So water is going to move from high concentration to low. All right, so that's going to be leaving the cell. This is a really bad situation for most any kind of cell. If it dries out, then it's not going to stay viable. So that's why if we're trying to preserve food, we're trying to keep bacteria, say, from rotting it, or growing on it, then we're going to try to dehydrate those bacterial cells. So we're going to add salt to our food or sugar to our food to preserve it. That's to, again, draw the water out of the bacterial cells. In a hypotonic solution, then we've got lower solutes, we'll say salt, for example. We've got a high concentration of water outside. All right, so water is going to be moving into the cell. This is not a problem for a bacterial cell because it has a nice intact cell wall. Right? If something happens to the integrity of the cell wall, like there's some antibiotics in the medium or something that makes that cell wall kind of weak, then yes, it's susceptible to lysing like a cell without a cell wall might, like an animal cell. Okay. So bacteria typically like maybe slightly hypotonic medium or isotonic. All right. These active processes, so remember transport, means that ATP is required in order to transport something inside the cell. So active transports, I'm not really too concerned about this terminology. You know, sometimes it's going to be more energetically advantageous for the cell to let something diffuse out, and that's going to help something else come in, or you know, a couple things coming in at the same time, just to make it energetically more favorable and, you know, use less ATP. Okay, and usually it's going to require some kind of a protein in the cell membrane there. And there's one kind of active transport that I'll draw attention to because this is unique to prokaryotes. And I'm not exactly sure why that is, why eukaryotes can't do this, but it's called group translocation. And this is when the substance that's being transported into the cell, like, for example, glucose, is changed during the transport. So the best example we have of this is when glucose is brought into the cell, it's often phosphorylated. Okay, a phosphate is added, so now glucose is changed to glucose 6-phosphate. You may recognize that. That's the first step in glycolysis. Okay, that is such a great system because if there's lots of glucose inside of the cell, okay, but there's, you know, a little bit outside, if there's a lot more of it inside of the cell, it's once we bring it in, it's just going to diffuse back out again because it's going from greater concentration to less concentration. We want to keep that, or the bacterial cell wants to keep that inside of the cell. So if it changes it, now it's glucose 6-phosphate, it can't leave again. It can't pass right through those same channels, and it's maybe a little bit too big to just diffuse out. So change it so we can concentrate it inside the cell and not lose it again. Okay, so that's, there may be other examples of translocation. This is just kind of the most common one that's always brought up. All right, so moving further into the cell, we come to the cytoplasm. There's not a lot to say about the cytoplasm, really. Part of the cell contained by the cell membrane. I was kind of surprised to learn not that long ago that cytosol is not exactly the same thing as cytoplasm. Cytosol is the part of the cytoplasm that's liquid. Okay, so that's the fluid present inside the cell. Okay, and what's in that fluid? Well, there's it's mostly water, but we might have some dissolved ions, some proteins in there. So cytosol is part of the cytoplasm. And then we have the nucleoid region of the cytoplasm, where the DNA is going to be located. So remember prokaryotic cells don't have a membrane-bound nucleus, but they do have a kind of specialized region in their cytoplasm where they keep all their DNA, and that's going to be in the form of a chromosome. There's usually one or two, most commonly one or two, and they are usually going to be circular, and they're going to be haploid. Okay, so our chromosomes, remember, we have 23 pairs, and they're linear. All right, so much different strategy for storing the DNA here. Most bacteria are also going to have numerous plasmids. Many. These are basically shorter chunks of DNA. Once again, they are circular. Usually the DNA on plasmids is going to encode for things that are not absolutely essential to the cell's survival. So you might have things like antibiotic resistance encoded for by a plasmid. Something like an enzyme in glycolysis is not going to be on a plasmid, it's going to be on the chromosome. We'll talk a little bit more about DNA when we get to our genetics unit. Okay, and the other significant structure in that cytoplasm is the ribosomes. Okay, of course, this is the site of protein synthesis. Okay, that's where the messenger RNA gets read, and proteins, amino acids come together to form proteins. Okay, the ribosomes are made of ribosomal RNA and proteins, and there's two subunits. Okay, so for instance, I just thought it'd be a good idea to compare the prokaryotic and the eukaryotic ribosomes at the same time here. Prokaryotes have a large subunit and a small subunit. The large subunit this shows the pieces of ribosomal RNA in there. It all adds up to subunits that we consider to be 50S. Okay, that S, it's measured in Svedbergs. Okay, which is a measure of the sedimentation rate. So if you broke up a cell and spun those cellular contents in a centrifuge, the rate at which they sink to the bottom, that's the measure Svedbergs. So it's really a combination of the weight and the shape, the size of the ribosome, just kind of a combination of all those things. Okay, so when we have a 50S, large subunit and a 30S small subunit, bring those together and it's 70S. Yes, it doesn't add up, but again, it's this measure of sedimentation rate. And then of course, eukaryotes, the large subunit is 60S, the small is 40, and that's up to 80. All right, so slightly different ribosomal structure between prokaryotes and eukaryotes. It's really a good thing. We can take advantage of that because we have a lot of antibiotics that will target the prokaryotic ribosome and not so much the eukaryotes. So it's a really good thing for us to kind of capitalize on. Archaeal ribosomes are very similar the prokaryotic ones, they have some different kind of lobes and things that are kind of more like the eukaryotic ones, but they have the same size, the same sedimentation rate as the prokaryotic ones. And of course, when we sequenced that ribosomal RNA in there, we found differences in the sequence between the archaea and the bacteria. Okay. Another structure, a couple of structures that may be present in prokaryotic cells. One is the endospore. So we mentioned this briefly a lecture or two ago when we were talking about identifying organisms and how whether an organism can make an endospore or not. That's a kind of a really nice way to put it in its own group. Endospores are not very common in the bacterial world. There's two main genera that are able to make endospores. That's the Clostridia and the Bacillus. A lot of really cool organisms in these genera. Bacillus anthracis, that's the causative agent of anthrax. A lot of scary Clostridium, the causative agent of tetanus, botulism, C. diff, Clostridium difficile, that's the Clostridium. So these are all spore-forming organisms. They're a little harder to kill because they can make this kind of hunkered down version of themselves. Right? This is a survival strategy. So what we think happens if you look at kind of the life cycle of the spore-forming organism, all these, the Clostridia and Bacillus, and there are a few other genera as well that are kind of lesser known. But when they're metabolically active, we say that they're a vegetative cell. So when they're growing and dividing, doing all the things that they might normally do, they're a vegetative cell. And if they were to suddenly encounter, say, some boiling water, yeah, they would die right away, like any other, say, E. coli or Staphylococcus. But if they encounter some harsh environment rather gradually, say for example, their nutrients are depleted, or maybe radiation levels are increasing, they can go through this formulation process. It takes between 6 and 10 hours. So it takes some time. There's about 20 unique genes that they have to do to be able to do this. They'll replicate their DNA, and slowly this spore coat will form around the DNA. Lots and lots of different layers of proteins and all sorts of things. Till eventually that vegetative cell falls off, and you've got a free endospore, which at this point can basically just sit for hundreds, maybe thousands of years and still be viable if the conditions were improved. They found endospores off of Egyptian mummies that I think have been thousands of years old, and they've been able to germinate them. So survival feature. We think that maybe if you have a medium of, say, a culture of bacillus cells, at any one time, some of the cells are going through this spore cycle. And then if some harsh condition does come on very quickly, if there are spores in that medium, those can kind of hang out until the conditions improve. Okay. Okay, so again, not reproductive, that's going to be a survival mechanism. So later on, we kind of push this to the end of the semester, but if you're in lab, you'll get to do a spore stain where the spores stain green, the vegetative cells stain red. It's always kind of a cool stain to see. If you just gram stained these organisms, they're gram positive, so you get these purple rods. The spores don't show up very well. We actually heat them to break them down a little bit to get the dye to stick to them. So again, if you're in lab, you hopefully get to that at the end of the semester. Just one other internal structure to mention that may be present. Some organisms may be able to make some kind of inclusion. And this is just a storage mechanism. So it could be some kind of lipid or starch, something else that is to the advantage of the cell to concentrate. A couple of examples of this,
gas vesicles. So
some photosynthetic bacteria maybe to their advantage to store up little vesicles of gas so that they can situate themselves in the water column in an appropriate way so they get the right wavelength of light, float up or down. Another really interesting example is hydroxybutyrate. And this is a starch polymer that again, some bacteria are able to concentrate in their cell. And remember, this is not a membrane-bound thing. There's a kind of a little proteinaceous layer around there, not a membrane, so it's not an organelle. But they can kind of concentrate this in their cell to use these nutrients for later. And we can manipulate bacteria by growing them with certain nutrients to make a whole bunch of this PHB. And we've been able to extract that from cells and use it for biodegradable plastic. So I didn't. Where's my picture? I left my picture off. But I have this picture of, maybe I posted it instead of putting it on mine. But you can make biodegradable plastic out of this PHB extracted from bacteria. And what's special about it, you throw this in the landfill, cover it up, and within a couple of weeks, it's basically degraded to nothing. So instead of the plastic that we use, from oil, that stays around for centuries till it biodegrades. So it's kind of cool thing when you think about using bacteria for our good, manipulating bacteria to help us. All right, well, let's move on to some eukaryotic structures. We're going to spend a lot less time talking about eukaryotes. We're all kind of more about bacteria in this class. But some of them structures do become important when we think about immunology a little bit later on in the course here. Eukaryotes can also have a glycocalyx. Remember, this is something that we said almost all bacteria have. Almost many bacteria have. It's very common to have this slimy substance secreted outside of the cell. Well, eukaryotes can have this too. With the caveat that it is attached to the cell membrane. Of course, in bacteria, that's going to be anchored to the cell wall. All right. So in eukaryotes, typically only eukaryotes that are going to have a glycocalyx are ones that don't have a cell wall. So only eukaryotes without a cell wall. All right, so plants, fungi, not going to have a glycocalyx. Right? Some protozoa, some cells in our body might have a little bit of a glycocalyx, like cells in our digestive tract apparently have a bit of a glycocalyx. Okay, same kinds of functions though, or some similar functions. Attachment to each other, especially some protection from drying out, just like bacteria. This function is a little bit different, strengthening the cell surface. So that makes sense. These cells that don't have a cell wall, this is just going to strengthen their surface a little bit, give them a little bit of protection from, forces of osmosis as well. Right. The cell wall, again, we're not going to spend nearly as much time on the cell wall. Remember, only certain eukaryotes have a cell wall, like I just mentioned, plants, fungi, or algae. Okay, so the function of this is pretty much the same though. Support, strength, shape. The composition is quite varied. So we said for bacteria, they always have peptidoglycan in them. Remember, never peptidoglycan in eukaryotic cell walls. Fungi are most commonly going to have a polysaccharide called chitin in their cell walls. There's a little variation there, but that's a popular one. Plants often have cellulose, something right that we can't digest. And algae, it's just incredibly varied. So like things like your diatoms are going to have silicates in the cell wall, but there's all different kinds of sugars that we find there. Some algae have agar in their cell walls. Remember that solidifying agent that we use for solid media. Some protozoa will have a proteinaceous pellicle. Okay, but not really a cell wall. Okay, we just call that kind of a protein layer around it. All right, and you know, these are all polysaccharides, and pretty much that's all we're going to say about that. All right, likewise, the cell membrane, going to blow through that very quickly because it's lot similar to bacteria. In eukaryotes, they do have sterols for added support, like cholesterol. And we think about cholesterol as being bad, but that's something that helps to stabilize our membranes when it's hot, and it helps to keep them fluid when it's cold. So it is an essential part of eukaryotic membranes. Same function, semi-permeable barrier. An additional function that bacteria don't have as part of their cell membranes is that they can be reorganized a little bit. Eukaryotes that don't have cell walls can move their pseudopods. Okay, this is just, again, kind of a reorganization of their cell membranes. And I'm going to come back to this slide, but here's an example of an amoeba, of course, extending its pseudopods. You can do that in order to move, amoeboid motion, or it can extend pseudopods in order to acquire some nutrients. In this case, this amoeba is endocytosing or phagocytosing some paramecium. Okay, so we could say another function would be transport. Okay, we could call that transport endocytosis. If it's, we could call that, another word for that is engulfment. Or movement of particles into a cell. Let's see. Okay. And if that movement is of liquids, the correct name is penocytosis. Probably less familiar than the phagocytosis. And that's movement of solids. Okay, so two different kinds of transport there that, of course, we don't see in prokaryotes because they have a cell wall. Okay, it's rigid, they can't extend and engulf. If a prokaryote wants to try to break some large compound down for nutrients, it has to secrete enzymes to try to do that and then bring the smaller parts into the cell. Okay, and of course, exocytosis is the opposite. We're exporting substances. So usually wastes. So two kinds of transport that are unique to eukaryotes, endocytosis and exocytosis. And we find that a lot of viruses will, this is a strategy to getting viruses into eukaryotic cells. They'll trick our cells into endocytosing the viruses, and they gain entrance to our cells. Okay, so moving inside of the eukaryotic cell, some eukaryotes are going to have flagella. And remember we talked about bacteria, the flagella are considered an external structure. In eukaryotes, it's membrane bound, so we're going to consider it inside the cell. Okay, so The function of the flagella for eukaryotes is the same as for prokaryotes, but pretty much that's where the similarities end. Different structure, different way of movement for eukaryotes. The composition of the eukaryotic flagella is microtubules. All right, so the protein that makes that up is tubulin. Remember, for prokaryotes, it's flagellin, makes up the flagella. So different protein, and those microtubules are in a 9+2 arrangement. Or a 9+2 structure, okay? And that seems like a pretty odd way to describe anything, 9+2. But you'll see over here, and that might be kind of small for you to see on your picture, but the 9+2 arrangement refers to 9 pairs of microtubules surrounding the outside, and then two more in the center. So that's that 9+2 arrangement. And down in the basal body, where the filament is anchored in the membrane or wall, that arrangement changes into a 9+0 arrangement. That just means that there's 9 triplets and none in the center. I don't know the significance of that change, but again, the structure changes a little bit in that basal body. Okay, there's no hook like there was for the bacteria. So we just have a filament and a basal body. So if we look at the motion, I'll refer you to a couple of YouTube videos that I found. Motion is in a whip-like manner. Okay, so remember our prokaryotic flagella spun in circles. When you're looking at these videos real time, it's really hard to tell whether it's a whip-like motion or it's spinning. They kind of look like the same thing. But this first video, if you just watch at the very beginning, it's like 11 minutes long, but at the very beginning, there's this some kind of protozoa. It looks like some kind of euglena maybe, but it's got this real long flagella, just the end of it whips around, and it's weird. It moves in the direction of the flagella, which is kind of unexpected. Like, I don't understand the physics of that. So the direction of motion oftentimes is in the direction of flagella. I think that it can be either way, but it's kind of cool just to watch that. And the second video has a comparison of some prokaryotic, I think there's spirochetes, remember the helical bacteria, their motion compared to some sperm, which of course are eukaryotes with flagella. And again, it's kind of hard to tell the difference, but the eukaryotes, again, moving in this kind of whip-like manner. Cilia have the same composition as eukaryotic flagella. All right, so if we think about what kind of cells have cilia, remember it's only eukaryotes that are going to have cilia. This is a structure that's much shorter than flagella, a little bit stiffer. Typically, the cell would have many of them on the surface. Okay. So what kind of cells have cilia? Protozoa. And I would say also a few types of animal cells. All right, we have ciliated cells lining our respiratory tract. And that cilia, its function, remember, moves in a whip-like manner to dispel debris out of our respiratory tract. Okay, if you are a smoker, your cilia don't work quite as well, so that leaves you a little more susceptible to any kind of respiratory infection. Okay, but this when I think about the movement of cilia, that's how I remember the eukaryotes move in a whip-like manner, because if all of these cilia were moving in circles, like how would that cell move anywhere? Obviously, our cilia in our respiratory tract is not moving in circles, it's moving to push things out. So hopefully that helps you remember which rotates and which moves in a whip-like manner. All right, so the organelles, I really kind of cut back talking about the eukaryotic organelles. I really just want you to have like a basic, what is this organelle? What is this organelle? And like we're talking really basic. Some of a few things I'll highlight that we're going to talk a bit later on when we talk about immunology. But you know what a nucleus is. That's where the DNA is stored. The ER, it's kind of a transport system. The rough ER has ribosomes. And what kind of proteins are made of those ribosomes? Ones that are going to get transported outside of the cell. So for instance, a cell that makes antibodies, that's a protein that gets secreted outside of the cell. That's going to be made on the rough ER, not on the free ribosomes. The smooth ER doesn't have ribosomes. There's a variety of functions in the smooth ER. One of them is synthesize lipids. The Golgi complex is kind of a further transportation, modification of macromolecules. For example, a protein that's made in the rough ER then might get sugar added to it, so it becomes a glycoprotein in the Golgi apparatus. Okay, and then two organelles that we're going to talk a little bit more about. Mitochondria, of course, that's where ATP is synthesized. A lot of metabolism taking place there in the eukaryotic cell. And chloroplast, if the cell is photosynthetic, then that's the sites of light harvesting. All right, so you can read more about it in your book if you want, but we're just talking basic functions there. The other organelle that I want to spend another minute on are the lysosomes. Okay, this is an organelle that contains digestive enzymes, and this is going to be important later on when we talk more about phagocytosis, right? Heart cells of our innate immunity, that's one of their main jobs, to fight invaders, is to phagocytize, say, a bacterium. So our cells are going to endocytose, say, some bacterium, say this is one of our white blood cells maybe. The lysosomes are, like I said, a sack of digestive enzymes that probably originally budded off from the Golgi. Those lysosomes fuse with the endosome that has that invader in it. 20 or so different digestive enzymes in the lysosome that work in a variety of different ways that usually end up able to kill that invader, and then, they exocytose, whatever's left over. Okay, so especially important in phagocytic cells, and we'll be talking more about that later. I want to end with a theory that's really gained a lot of acceptance ever since it was proposed by Lynn Margolis in the 1970s. When she first proposed this theory as to how eukaryotic cells originated, she basically got laughed out of town. People made fun of her at when she presented papers and things like that. But over the years, it makes more and more sense. We'll talk about kind of why we think it makes sense. It's not something we can test because it's an origin theory. But again, let's see. Let me write this a little more succinctly. So this is a theory that explains how eukaryotic cells evolved from prokaryotic cells. Okay. And also how those organelles, the mitochondria, and the chloroplast came to be within a eukaryotic cell. All right, so the theory goes is that early in the history of the Earth, right, we think that prokaryotes were the first life form, and there was some anaerobic larger prokaryotic cell that engulfed an aerobic smaller prokaryotic cell. And instead of that larger cell chewing up the smaller cell and using it for nutrients, somehow that smaller cell was allowed to live inside the larger one as an endosymbiont. So an endosymbiont is an endosymbiosis is when you have two organisms kind of living together, and that smaller organism lost the ability to live without the larger one. Okay, so that aerobic prokaryote allowed that anaerobic prokaryote to respire using oxygen and maybe get more energy that way. So at least that's the theory. And eventually this endosymbiont, became acquired, more organelles and is what we would call a eukaryotic cell today. All right, so support for this theory. Mitochondria and chloroplasts they actually have their own DNA. Okay, that's not the case with other organelles, like lysosomes or ribosomal RNA, but they have their own DNA, and they actually divide independently of the rest of the cell. So they're kind of on their own timetable, they have their own DNA. The other very interesting thing is that mitochondria and chloroplasts have their own ribosomes. And they have 70S ribosomes. All right, so that's to me the most intriguing thing. They've got prokaryotic ribosomes. All right. So that's kind of interesting. And of course, the ribosomal RNA in them is similar to that of bacteria. Okay. And also mitochondria and chloroplasts are about the same size as prokaryotes. Right, about a micrometer. All right. So all of those are evidence for the theory. Not everything matches up. Like a lot of the DNA that is actually necessary for the mitochondria and chloroplasts to replicate is now located in the nucleus. So we think, well, did it migrate there, or why is that the case? And not everything matches up, but kind of I would explain why mitochondria and chloroplasts have their own ribosomes. Okay. All right. Questions? Anything about structure? Of prokaryotes or eukaryotes? All right. Once again, I had a prettier picture that I somehow left off of here. Oh well. So we'll move on to chapter 12, which we are going to kind of touch on. Definitely not hitting all of this chapter. The questions that I came up with here are not very creative. But we're going to start out with this is really our last time that we talked very much about eukaryotes at all, other than talking about some of our own immune cells. But I want to make sure that you know the difference between algae, a protozoa, and a fungi. So what are some of those characteristics that we look for and say, oh yeah, we would classify this as in one of those groups? I want to talk a little bit about one of our protozoa, and that's a organism that causes malaria. And then we're going to look at some characteristics of fungi, why we study it, some terminology that we use to describe fungal structures. But I've cut a little bit of that material on fungi, like the whole reproduction stuff. So you can thank me at some point for that. So if we think about classifying eukaryotes, so of course, remember Linnaeus back in the 1700s, he classified things as either plants or animals. We've been revising that ever since. Remember I talked about that Whitaker system that split into kingdoms, the fungi, the protists, the plants and animals, the monera, or what they call bacteria. We now know with that ribosomal RNA sequencing or now we consider actually there to be many different kingdoms here. So technically, when I use the term protozoa, that's not really an accepted classification anymore, but it's still in use because that's what people recognize. Like I couldn't even rattle off more than a couple of these different kingdoms down here. So we still use this term protozoa. Your books still use it, or protista. But just recognize that not all organisms are going to fit nicely into either being a protozoa or a fungi. For example, this Stramenopila, those organisms include the one that caused the great potato blight in Ireland a few centuries ago. They're actually called slime molds. So they're kind of like fungi, but they're kind of like algae as well. So they just, not everything's going to fit nicely into either protozoa, fungi, or algae. All right, but let's look at some characteristics that the protozoa do share. I should mention that chapter 12 starts out with a big review of mitosis and meiosis. You can skip that. We don't, if I went into the fungal replication, then you would need to know kind of a basic idea of mitosis and meiosis, but we're not going to cover that. So, read if you're interested, otherwise you can skip over that. Characteristics shared by protozoa. So start with the basics. Remember, they're eukaryotes and they're single-celled. Sometimes students memorize all these minutiae and then they kind of forget the whole big picture of things. So don't forget that. They, like I mentioned a little bit ago, they wax cell walls. They often have a protein pellicle. And most are motile. And in fact, we often will categorize them based on their kind of motility. Do they use pseudopods and move like amoeba? Do they have cilia? Do they have flagella? Let's see. In terms of how they get their nutrients, most are chemoheterotrophs. And that's a term that we are going to pick apart more in the next lecture. But basically, this means that they eat stuff. Okay, we are chemoheterotrophs as well. Trophs means to feed, and the prefixes there basically describe how they get their energy and their carbon source. But some are photosynthetic, which we would call them photoautotrophs. Okay, so again, most of them just eat nutrients. They get their nutrients from organic sources. There are a few that are photosynthetic. Many are free living. In fact, we would describe them as being ubiquitous in nature. Pretty much anywhere, any moist place, there's going to be protozoa living there. But there are some that are considered parasites. Okay, if a virus or a bacteria causes a disease, we call them pathogens. If a protozoa causes disease or a worm or whatever, we call them parasites. So just a little terminology there. So there are certainly some infections caused by protozoa. Here's just a couple examples here. Trypanosomes, they cause African sleeping sickness. This is a picture of a giardia. Odd looking. This is a single cell, but it has two nuclei, kind of look like eyes there. This is the reason that you don't drink water out of a stream, no matter how clear it looks. Because there could be protozoa in there, like giardia or cryptosporidium, that would give you a nasty case of diarrhea. Okay. And I want to talk about just a couple of structures that we see in the life cycles of protozoa. And your picture, I think, does not have the picture there. Somehow I was looking at that the other day, and I was like, oh, I like to have a graphic whenever I can, and I left that out. So sorry, it's just a couple of terms here. It illustrates all protozoa have part of their life cycle that's considered a trophozoite. Okay, and that just means the motile feeding stage. Okay, so lots of varied life cycles, but they all have a motile feeding stage. So this graphic came from a website that was talking about that amoeba that you may have heard of, that you may have heard of somebody going swimming in a freshwater lake, and they died a few weeks later. So this causes a meningitis. It migrates through somebody's nose that's been swimming in fresh water, and basically they don't figure out what's wrong with them until they do the autopsy. Right? Very rare. It's called Nigeria fowleri. Again, it's a very rare protozoa. This has a protozoite stage that's usually an amoeba. Sometimes it switches to the flagellated form. That's the motile feeding stage. And it can also cycle between a cyst stage. So some bacteria can what we say encyst. Okay, and the cyst is let's see, a hardy resting stage. So more typically, if a protozoa finds that its source of water is drying up, it could encyst in order to not dehydrate. Okay, and then when water is present again, it can change back into its motile feeding stage. So really, those are just the only two terms that I would say. This was what you would need to know about any protozoa life cycle. I want to talk a little bit more, just mostly because, hey, let's talk about at least one parasitic protozoa. Let's talk a little bit about malaria, just because it's so fascinating to me. And this is the number one threat of Americans traveling to other countries in terms of their health risk. It's the number one threat, malaria. Okay. Right now we don't have malaria in this country, but we have in the past. So it's only because we did such a good job of wiping out all the mosquitoes that we got rid of malaria. Those mosquitoes that carry malaria are back in the U.S. and we could have this transmitted again in the U.S., especially with climate change. What causes malaria? There are five different species of this protozoa, Plasmodium, that cause malaria. So here's a picture of the falciparum Plasmodium. And you don't need to memorize those, but just to point out that there are different nuances in malaria. There's different timing depending on which protozoa you're infected with. It might be a different time to come down with symptoms, and your symptoms are definitely going to be different depending on what kind you have. This falciparum is generally considered to be the worst kind to get. That's the kind that's endemic to sub-Saharan Africa. And then typically it's the most severe, causes the most deaths. The Plasmodium vivax is probably the second worst. That's the one that's in Latin America and a lot of Asia. The Plasmodium malaria and ovule are very small percentage of cases of malaria, and this Plasmodium knowlesi, actually there's only been a couple cases of that. That's a species that has been known to infect gorillas for a long time, and only recently been realized that this could be in humans as well. All right, so this Plasmodium doesn't harm the mosquito as well at all. So it's the female Anopheles mosquitoes. They're the only ones that take blood meals. They need it to for nutrition for their eggs. The males don't bite us. Okay, so the life cycle of the Plasmodium cycles between the mosquito and us. Okay, so we can't give it to each other unless, I don't know, maybe if we share blood somehow. But you need the mosquito as part of the life cycle. And what do the disease manifestations look like? Typically, it cycles through three different stages. Each stage might last like 6 to 10 hours, and then every couple days you'll cycle through it again. So the patient, maybe a couple weeks after they're bit with an infected mosquito, they go through a cold stage. So chills, shivering, shaking. And then maybe 6 to 10 hours later, they go into the hot stage. So high fever, achiness, headache. And then another 6 to 10 hours later, that fever breaks, and we get the sweating stage. Patient gets the sweats, and they're very tired. Okay, fever comes back down, they're sweaty and they're tired. So to me, I read that and I think, oh, that doesn't really sound that bad. I mean, maybe it sounds like the flu. Well, I happened upon the Gates. Bill Gates, I couldn't think of his first name. Bill Gates has done a lot of work to try to fight the spread of malaria. And if you go on his website, Gates Notes, he has a description that somebody wrote of what it felt like to have malaria. So I awoke to it felt like lightning going through my legs and then spreading through my body and head. Probably the worst headache, body aches, and chills you could possibly imagine. It felt like I was being stung repeatedly by an electric shotgun and could barely control my movements. The pain was so intense, I actually believed I was dying. So it sounds pretty nasty. There are other complications that we see. Anemia is very common because it's living in red blood cells, the Plasmodium. There can be neurological complications. I had a friend that came back from Africa and had malaria, and he was in a coma. We didn't think he was going to make it. So there's a lot of complications. Kidneys shut down sometimes. So adults tend to build up a little bit of immunity to it if they're living in a place that has malaria present all the time. It's much harder on children, higher mortality in kids. About on the order of 500,000 people die worldwide of malaria every year. So it's really a tough thing. It is treatable. I'm not going to get into the treatment so much, especially when you catch it early on. It's treatable. There's a lot of resistance spreading among the Plasmodium to the drugs that we're using. The drugs that you have to go on to try to keep from getting malaria, a lot of side effects, nausea and stuff. So that's not ideal either. So it's really will continue to be a tough thing to deal with. I wanted to. This is a pretty intricate map of the life cycle of a Plasmodium, and I just wanted to point out a couple things that we maybe should know from this. We didn't understand the life cycle of Plasmodium until like the early 1900s. Sir Ronald Ross was the one that elucidated that, or is given credit for that. So it's very complicated. But if we start right here, the number one, where a mosquito takes a blood meal. So a mosquito that is infected with some kind of Plasmodium bites a human. And they have actually like 4 or 5 different proboscises that they stick into us. Of course, one, they're drawing out our blood. With another one of those proboscis, they are injecting their saliva, which has an anticoagulant in it. And also in their saliva, that's where you'll find the Plasmodium. Right? So that gets injected into the human. First of all, it infects the liver. Okay, so that
plasmodium
migrates to the
liver,
and the liver cells. There's some different ways we describe
that cycle in there.
But at some point, then it moves into the red blood cells. Okay, and this is where it's causing the symptoms in humans. And this is also, you'll see, that trophozoite stage. So the motile feeding stage, as many as like 10% of the red blood cells can be affected by the plasmodium, and that's why we're getting the high fevers and the sweats and all that stuff. We won't get into the mating of the plasmodium there, but at this point, if another mosquito that's not infected takes a blood meal from the same person, now that mosquito becomes infected. There can be a cyst portion of the life cycle there within the mosquito. Okay. So extremely complicated life cycle. Just so you know that it infects our liver first and then the red blood cells. And again, cycles through those symptoms. Okay, so another I forgot. Turn. I had another thing that I wanted to post from that Gates notes website. So it talks about what's the most dangerous animal to humans. And you probably, like, first thing I thought of was a lion or maybe a hippopotamus in terms of how many deaths they cause. Well, actually, in terms of the deaths caused of an animal to a human, snakes, you get like 50,000 deaths a year from snakes. Dogs is a little bit less than that because they pass rabies. Humans kill a lot of other humans, maybe like 400,000 a year. But above that, mosquitoes is by far the greatest threat to human life. His number is about 750,000 people a year are dying because of, again, infections that mosquitoes pass around. There's about 14 different infections that mosquitoes can give us. Okay, the only protozoa is the malaria, all the rest of them are viral. Yellow fever, chikungunya, dengue, Zika, Eastern equine encephalitis, and there's a bunch of more encephalopathies. So it's just sort of interesting if they made the point that if we got rid of mosquitoes, totally wiped them out, we'd be a lot better off. We think that there's a lot of animals that eat mosquitoes, of course, but they eat other things as well, and think that we would be fine if we could totally get rid of them. But all right, so anyway, I forgot to post that. Sorry about that. Or show you that. Algae. Moving on to other eukaryotes. I don't have a lot to say about algae because they don't really cause disease. Maybe a red tide now and then, some toxins from a red tide, but in general, the characteristics, of course, are eukaryotic and photosynthetic. They can be unicellular, multicellular, or colonial. So something like a diatom here, of course, is unicellular. This is volvox, which are considered a colonial algae. And then the multicellular ones, here we've got our red algae here at the top. That's what we extract agar from. And giant kelp. So this is a plant, a seaweed that grows for miles and miles in the ocean. It's considered an algae. So if you think about what's the difference between plants and algae, it's not just that plants are big and algae are small. It actually has to do with the reproductive structures. So in algae, every cell in the reproductive structure can become a gamete, and that's not the case with plants. So it's a little bit more complicated. What would you consider algae? What you would consider a plant, not just big and little. And we're not really going to go through those cycles, so you don't need to worry about anything more specific. What about plankton? What would you consider plankton?
I had a picture of plankton from SpongeBob that I was going to show you. What would you consider that? Plankton are actually defined by ecological niche. So any unicellular organism that lives in a large body of water is considered plankton. Okay, so that could be Protozoa, that could be bacteria, that could be archaea, it could be algae, all those things could be considered plankton. Just in case you were wondering, I like to throw out interesting things to talk about at the dinner table. All right, we'll just begin our fungi, and I'll finish it with our next lecture. But thinking about primary characteristics of fungi. Of course, we talked about they have a cell wall, often composed of chitin. Cell wall. And the other major characteristic is that they are not photosynthetic. Right, so you've got something that's photosynthetic, that could be a plant or an algae or a protozoa or even a bacteria or archaea, but not fungi by definition. All right, so if you think a little bit about, well, why should we care about fungi? You can probably think of a lot of ways that fungi affect us. A primary thing that they do for us, they are nutrient recyclers. Okay, they decompose dead trees, for example, dead animals in the woods. Okay, nutrient recyclers. Okay, we use fungi for food, drink. They yeast are fungi that are responsible for alcoholic fermentations. We have antibiotics, drugs that are secreted by molds. Okay, penicillin comes from a penicillium mold. Also, a symbiotic relationship or symbiosis with plants. We know that this, Plants. We know now that this is really common, that most plants have some kind of symbiotic relationship with fungi in their roots. Because the fungi will live near the roots and help the plant roots absorb water. Okay, so we call that relationship myco. Let me spell this right. Mycorrhizae. Okay, so that's probably I'm thinking an unfamiliar term. So mycorrhizae, again, this is a fungal relationship with plant roots. Again, helps them absorb water. So I got this picture up right here from some, it was an ad for some kind of lawn chemical that was supposed to encourage mycorrhizae. So here on the left, it has a clump of grass that has lots of these mycorrhizae. Again, fungi growing with the grass roots. And then a clump that doesn't have that relationship. So I guess you're supposed to say, oh, wow, look at this great green grass here, and this is, less vigorous in some way. But we think that again, helps our crops grow, and that's something that we often fail to recognize. All right, if we start looking at some of our structures that we find in a lot of fungi. So we'll start with the molds and fleshy fungi that's fungi like mushrooms. Okay. A lot of times the mushrooms will have these structures underground. All right. So all of the molds have hyphae. These are filaments of cells.
And we have a couple ways that we describe those hyphae. Okay,
either they're septate,
and that means that they have cross walls. So basically the filament is kind of broken up into cells. You can't see very well from their picture over here, but there are divisions between these cells here, cross walls, little pores in the cross walls so the nutrients and water can flow along the whole hyphae. And then some hyphae are considered aseptate. And that just means that they have no cross walls. Basically, the filament is just kind of one big cell, multinucleate. So it's considered cenocytic. And that's just a term that means multinucleate. Okay, so cenocytic, aseptic, aseptate, not aseptic, aseptate hyphae. Okay, and then we also consider our hyphae either vegetative or aerial. Okay, so just to finish up here, the vegetative hyphae their job is to obtain nutrients. And the aerial hyphae are reproductive. Okay, and I'll show you some other pictures of those very soon. The vegetative hyphae, you get a big mass of these, and it's called mycelium. Okay, so get that term down, and I'll illustrate that a little bit more next time. Looks like, yeah, I got my wrong set of slides up here. I left off some pictures that I meant to show you. So okay, look for my lecture posted hopefully sometime tomorrow. Email me with any questions. I'll get some practice questions out later on this week once I get the test finalized for next week. So okay, have a good day.